Over the last 10 years, there were over 100 outbreaks of foodborne illness caused by Salmonella, E. coli, and Listeria, and linked to the consumption of leafy greens, sprouts, tomatoes, and other vegetables. In the marketplace, it's difficult to distinguish between the vegetables that are infected with Salmonella and those that don't contain it. Less than 0.1% of produce may contain human pathogens, but still it's important to eliminate those sources of infection in the production chain. We focused on the impact of the fertilization, irrigation regimes, weather conditions. We also looked at the cultivar selection as potential production practices that may reduce susceptibility of tomatoes to contamination with Salmonella. We also learned that different tomato varieties responded differently to the levels of fertilization with nitrogen, potassium, and also irrigation regimes. So in designing food safety strategies, it's important to take into consideration not only the levels of irrigation, fertilization, but also the crop genotype. We have conducted this study in 3 seasons. And what we learned is that tomatoes that were harvested in the driest, sunniest seasons were most susceptible to Salmonella contamination. We suspect that in the driest, sunniest times of the year, the native microorganisms that may be present on tomatoes and are typically not pathogenic may be reduced or killed off by the sunlight. And this creates potentially an opening for human pathogens like Salmonella to gain entry into the tomato fruit. We also find that green tomatoes are significantly more resistant to Salmonella infection. However, red ripe tomatoes may be much more conducive to Salmonella proliferation. In terms of the fertilization and irrigation regimes, we find that it's important to modify them in accordance with the tomato cultivar and also with the weather conditions. We're excited about this research because in the long run, we want to promote food safety by modifying easy production practices like irrigation, fertilization regimes, cultivar selection, and maybe looking for the plant genotypes that will be more resistant to Salmonella. If you want to find out if the produce contains Salmonella, it's important to submit it for microbiological testing in the commercial specialized certified laboratory. In the commercial laboratory, samples will be processed and they'll be plated on a selective medium. The selective medium contain pH indicators, and they also contain specific nutrients that can be utilized by human pathogens. And the utilization of these nutrients will affect the color of the colonies and the plates. On these plates, Salmonella forms black colonies, and fecal coliforms form yellow colonies because of their ability to utilize certain carbon sources and then change the pH of the medium that can be easily detectable. However, please keep in mind that only 0.1% of produce in the marketplace may be contaminated with the Salmonella or Listeria or E. coli, and the vast majority of the produce in the supermarket is completely safe and free of contamination.